All right, well, we might make a start. So, good morning and good evening, everybody. My name is Steve Meikle. I'm at the University of Sydney, and I'd like to begin by recognising the Indigenous custodians of the lands on which the University of Sydney sits, and those are the many different lands you are joining us from today. Today, I'm on Gadigal country, and I pay my respect to elders for their leadership in maintaining a continuous exchange of knowledge and customs across thousands of generations for the benefit of all of us and those who follow in their footsteps. So today we'll depart from our usual webinar format. It'll be in the style of a conversation with two standout leaders of our field, the two people who are most responsible for bringing total body PET from a concept to reality. They are, of course, Professors Simon Cherry and Ramsey Badawi from UC Davis, and they will be joined in conversation with Alex Burton, who is the marketing and outreach lead for the Australian National Imaging Facility. So Simon and Ramsey both hail from England, and they both studied physics and astronomy. Like the other famous English pair, Lennon and McCartney, they left the shores of England to see if they could make their mark on the USA and the world, which of course they duly did. So Simon is Distinguished Professor Emeritus in Biomedical Engineering and Radiology at UC Davis, and Ramsey is Professor of Biomedical Engineering and Radiology and Co-Director of the Explorer Molecular Imaging Centre at UC Davis. They've been working together at UC Davis for about 20 years and have co-authored more than 60 publications, including several recent, very highly cited papers on total body PET. You could say they are the Lennon and McCartney of PET. But how does their collaboration work? Do they write the material together, or are they more like an alternating double act? Have they let fame and glory affect their relationship? We will find out all this and more as Alex interrogates them over the next 40 minutes. Over to you, Alex.
Thank you very much, Steve. And it's a pleasure to meet you, Professor Cherry and Professor Badawi. Thank you both. On behalf of National Imaging Facility, I'd like to start by saying how honoured we are to be joined by such esteemed guests. We're also joined by hundreds of people across Australia and internationally today, and I welcome you all to this special session of the Australian National Total Body PET webinar series. So I'd like to open the session with one of the questions I think we'd really love to hear firsthand. What initially inspired you to pursue a career in biomedical imaging research? And maybe I could start with you, Professor Cherry.
Sure. Well, first of all, Steve, that was one of the funniest introductions I've ever had, I think. And so I'm not sure we can quite live up to Lennon and McCartney. Certainly we're not paid as well as they were. But anyway, so my background physics as well, and as I was trying to decide what to do at the end of my degree, I sort of accidentally stumbled across medical imaging actually as a field. I saw some ads for some PhD projects, and it seemed like a good combination. Imaging, which I loved from my astronomy background, nuclear physics, which I also loved, and also potential direct benefit to humanity. So what's not to like about that? So that's sort of how I got into medical physics.
And I'd really like to hear your side of the story, Professor Badawi.
So mine is a truly mundane story, actually. I fell into it completely by accident, to be honest. So I was working as a systems administrator at the first clinical PET facility in London in the UK as well, about 1991, something like that. So really I was just futzing around with the computers and doing networking and making sure there was enough disk space. And the physicist at the centre, Dr. Paul Marsden, this was back a long time ago when we were both much younger, and Paul was still quite early in his career, and he just reached the point in his career where he needed a PhD student. So we were at a staff meeting, and he said, you know, I want to get a PhD student, and I just stuck my hand up and said, oh, I'll do that. I was getting a little bored with being systems administrator, and so this seemed like a fun thing to try and do. And so, and for some reason, Paul said, yeah, all right then. And we wrote a little grant together to get some funds to do it. And that's it. That's how I ended up in the field. So there wasn't any strategy, wasn't any grand vision, just the opportunity presented itself and I grasped it. And I'm very glad, of course, that I have done. It's been a tremendous journey.
There's nothing like a spur of the moment decision to kickstart a career. But it sounds like your backgrounds could really complement each other in this journey. And as Steve mentioned, obviously, it's been a huge collaborative project to develop Explorer, the world's first total body PET scanner. How did this collaboration come about, and had you worked together previously, or was it very much a strategic collaboration? Ramsey, would you like to take this question first?
Yeah, sure. So the story goes back quite a way to not long after my story began in medical imaging. So I'd heard about Simon from my PhD advisor, because my PhD advisor was postdoc in the lab that Simon was doing his PhD. There's a lot of stuff. You know, I have bits of Steve Meikle's code in my PhD as well. Actually, there's a lot of exchange that went on from this gang. Anyway, so Simon was a good friend of my PhD advisor, and I was doing my PhD, and those of us who've done PhDs will be familiar with, sometimes you can go through a real dip in motivation and you wonder why you're doing it and everything seems to be going wrong and you're sort of faced with despair and you're just thinking, how do I just keep going? And I was going through my phase of doing that. I think everybody I know who's done a PhD has gone through that. And my wife, who's not in the field, she said, look, we're going to California at Christmas. And Simon was at UCLA at that time. She said, why don't you go and see Simon? Why Simon's name came to her mind, I don't know. But she said this, and I thought, that's not a bad idea. And so we arranged it. And Simon kindly let me visit his lab and hang out with the scientists he was collaborating with. And I went and visited Simon in his office, and we sat in his office. And Simon has a knack of inspiring people. And I came back out all fired up, going, yeah, this is great. And went back and finished my PhD, and it was all good. So I knew well in advance that Simon was a good guy. And then back, this would have been about 2003 or so, 2004, I was thinking about making a move. I was working on the East Coast of the US in Boston at the time, and I was thinking about making a move. And so I was making calls around my contacts, and I called up Simon to see if there was any opportunities going at UC Davis. And he told me there was a vacancy in radiology for a PET physicist. And so I applied for that, and sort of Simon helped guide me through that process, and I landed at UC Davis. So that's really how our collaboration started. I should point out that we were also involved the year before we'd started working together, organizing the IEEE Medical Imaging Conference together. So Simon was the scientific program chair, and he'd asked me, presumably having remembered a half-decent conversation back in UCLA, if I would be the deputy program director with him. So we'd already started working on quite a bunch of stuff. So on an operational basis, we already sort of knew how each other worked from that.
And you obviously had that trauma bonding of organizing a conference together.
Absolutely. Trauma bonding is a very good way of putting it.
We had lots of that.
Simon, would you like to add anything there? It sounds like you probably
there's not much to add. I mean, it's, I actually knew Ramsey when he was a PhD student. As he said, I visited King's College where he was at the time for a few weeks to work with his supervisor. And actually his supervisor, Paul Marsden, I think is somebody that's motivated both of us and influenced both of us in our career, actually. He really taught me everything I knew about instrumentation when I was a PhD student. So it's a nice parallel there that we've both been influenced by him. And the only other thing I'd say is, this dip of enthusiasm and motivation and challenges that you get during your PhD is just good practice for the rest of your academic career, because you keep hitting these barriers time and time again, and you need somebody to help push you through it. And Ramsey's often been the one to push me through those barriers as well since then. So, yeah.
It sounds like you very much inspire each other to keep going. But it's probably maybe not the best advertising for a career in research to tell everybody that you have a career of despair in front of them.
No, not all despair. It's ups and downs, right?
So do you have any memorable stories you'd be willing to share from when you first started collaborating with our audience?
Well, I remember the meeting in my office where we first talked about doing total body PET. And, it was a very pivotal moment, I think. You know, Ramsey had just got to UC Davis, and we were just chatting about what we might be able to do together, and you know, he'd been doing some really interesting simulation work to see what would happen if you started making PET scanners longer and longer. And you know, at the time, I think PET scanners were maybe 10 or 15 centimeters long, something like that, so really, really short. And he was going all the way up to 60, 70 centimeters, which seemed outrageous at the time, completely outrageous. And he was showing that things didn't deteriorate in terms of scatter fraction and random fractions as much as one might think. I was coming at this from a very different angle, doing a lot of instrumentation development, mostly around preclinical small animal imaging. And of course, we built total body mouse images, although we hadn't really realized what we could do with that. But we were used to imaging the whole organism with mice. And so as we were sort of thinking about areas that we could work together, it sort of naturally came out of this, well, let's think about actually building a longer human scanner. And, you know, not wanting to shortchange ourselves, we decided, you know, if we were going to do this, we should go the whole way and really try and build something that would cover the entire human body. Because that was exciting. Nobody had ever done that before, not with any medical imaging technique. So we thought that would be super cool. If we'd known the trouble we were going to get ourselves into, maybe we would have had second thoughts, but fortunately we were totally naive.
Would you say that was your eureka moment?
Yeah, I mean, it was certainly, you know, having somebody else that was as enthusiastic to do this as I felt at that moment was the validation. If it had just been me sitting on my own, I don't think I'd have done it, no.
It sounds like you really complement each other and you really inspire each other, and it's wonderful to hear. But obviously, you know, you just mentioned now that you faced quite a few challenges while you were developing total body PET, and I guess one of the major challenges in every field is taking an idea from concept right through to implementation. Could you tell us a little bit about your journey and some of your insights having taken total body PET from this sort of initiation and idea beyond implementation all the way through to having a clinical impact?
That's a bit of a 20-year question there. There's an awful lot to unpack. So I could talk a little bit about the technical side, but I guess the key thing in the early days was simply the challenge of trying to get funding for this idea. And what had happened after we'd had that initial conversation, Simon knew that he was up to give one of the plenary lectures at the Society for Nuclear Medicine meeting the following year. This would have been 2006, I think it was. And so, what was it, of mice and men, or from mice to men? I can't remember the exact.
Right, something like that.
Something like that. And you showed a graphic showing a total body PET scanner. Actually, if I remember right, it was embedded in a total body MR. The idea, certainly that was the first time that anybody in public had talked really about doing a total body PET. And it wasn't greeted with vast amount of enthusiasm, to be honest, as a concept. I think probably 20% of the people who heard that talk jumped up and down and thought, that's just a tremendous idea. And 80% of the people just said, don't be silly, take your Percocet, sit down and be quiet. You know, it's just too silly an idea. And I feel like also for a long time in the clinic, nuclear medicine had been a sort of Cinderella operation, didn't really make money for the hospital, didn't really make money for the department if it was part of the Department of Radiology. And by this time, PET-CT had taken off and it was becoming part of the radiology department, which was much more financially powerful. And there was a bit of sort of, I think in the field, there was, and I've talked about this before in some of these interviews, is a bit of Stockholm syndrome. We kind of didn't feel as nuclear medicine people that we deserved projects like this. When, of course, in MRI, these kinds of investments have been going on for a very long time. People, because obviously it's going to take $10, $20 million to put one of these things into reality, right? So in MRI, yes, people have spent that kind of money on instrumentation. Nobody would want to do that in nuclear medicine, right? Because it's too minor and too niche. So we had to overcome that, and that took a lot of time. Actually, it wasn't a totally trivial process. It meant a lot of elevator pitch conversations, a lot of bringing this up when we got a chance to talk in public, And I guess people, maybe Steve can answer this, people must have got a bit fed up with us actually, going on about the same idea year after year, because it was a matter of many years before we did get funding. I don't know if you want to add anything to that, Simon.
Well, I think that's right. You know, so much of the focus was on funding initially. We had no money, so all we could do is talk about it, which we did frequently, as Ramsey said. Another thing I want to point out is it's not so much that it was our idea. That idea had been out there for some time. You know, Terry Jones, who's a great friend and collaborator, had shown a slide at an IEEE conference back in 1990 or so, of two large panels covering the whole body. Various other people had talked about this concept. So it wasn't so much it was our idea, it was just our determination that we were going to try and do it. And as Ramsey said, I think getting over the sort of rather small town mentality of the field was a big challenge. But I think obviously the funding was a big challenge initially, and part of that was us not really having thought through the applications, both in the clinical space and in research, and being able to really articulate those, and for people to actually see where it was going to make a difference and how it would make a difference and how we would justify the enormous cost, because that's what everybody was thinking about. This is too big, it's too expensive, it's not going to make a big difference. And then, if we fast forward a little bit, we could spend the entire hour on this question. But the other thing that I always remember is that, a big moment when we got the big grant in 2015 to build the prototype, which was a very exciting day. I was really excited for about one day, until the next day I woke up and realized, oh my goodness, we have to build this thing. Everybody's looking at us. Everybody in the US thinks we've stolen all the grant money in the field for the next several years, because we got a large amount of money. The eyes of the world are upon us. We cannot fail. And here we are, a bunch of academics. We've never built anything on this scale. And so suddenly at least I realized that, now instead of worrying about money, now I was worrying about how on earth we were going to make this thing. And so the next period of time was trying to persuade a company to work with us. And that was another interesting experience, going around different boardrooms, talking to management of these big companies, trying to get them to work with us.
I remember Simon telling me, it's about that time, he said, you do realize, Ramsey, that one way or another, this is a career-ending project. Because if it failed, that was the last time anybody's going to trust us with any money. And if it succeeded, it'd probably be our meal ticket for the rest of our careers. And Simon, in fact, has managed to close out. Well, he's still, of course, going as an emeritus professor, but he has managed to retire at the top of his game with this project under his belt. I mean, the other thing we did have, sorry to interrupt, please go ahead.
You go ahead.
I was just going to say, we had some giant shoulders to stand upon, actually, because some groups had attempted to build these some things not quite on this scale, but getting there. So the Siemens CTI group had built this scanner, which I think was called the P39, which was a series of, I think, 5 or 6 flat panel detectors, which covered about a 50 centimeter axial field of view. And then Hamamatsu had also the SHR 92,000, I think was the name of the scanner, which was this giant 60 odd centimeter BGO scanner. And neither had been terribly successful. And from what we'd learned, a key part of that was that their electronics had not truly scaled with the magnitude of the problem here, because you can collect a ton of signal when you have a really long scanner. And if I remember rightly, the electronics was not too dissimilar to what was going on the conventional scanners, and they just simply couldn't handle the count rate. So the only kind of studies you could do were really low injected dose studies, and then you didn't get the benefits of the great signal that you could collect. So we had those lessons to learn from. But I think luckily the reviewers of our grant had not had the realization that Simon realized the day after we got the grant, because when I look at that grant now, and it said that we were going to get undergraduate students to wrap reflector material around all the little crystals that were supposed to go in the back of that thing. I mean, that's a fatal flaw from the get-go, really. And I can't believe they trusted us with the money when that was in the plan. But clearly it wasn't practical. We had to do something different. We needed the quality control that an industrial partner could bring to make sure this thing actually worked. Because you've got 8 times as many components here as you would have in a regular scanner.
So it sounds like taking people on the journey with you was as much of a challenge as anything else, and helping them to understand where the technology could be used, getting buy-in from, like you said, the academics, industry, and the clinicians all in one go, and then getting those people to share your vision was a major challenge for you guys. But I guess if you could rewind time and do something differently, what would it be? What were the lessons that you learned?
So many. I mean, first of all, I worked in preclinical imaging for a good decade and a half, doing total body imaging of mice, supposedly, but never thinking about total body applications. Always imaging tumors, response to treatment. We could have prototyped so much stuff for total body PET if I'd been thinking a little more clearly about the capability that was there. So that's a regret looking back, that we missed that opportunity. I guess, perseverance is another thing. Don't take no for an answer is another big message of this project. If you believe in it, obviously we had to do other things because we had to keep our labs running. So if we put all of our eggs in this basket, we would never have made it. So we had to have other projects going on that were funded during that time period. So that sort of speaks to having diversification of a research portfolio. So you can put some of your eggs in the high risk basket, but could still keep going for the length of time it takes. So those are a couple of things that I've learned. Ramsey, what about you?
Yeah, I mean, I feel like, the idea of rewinding and then doing it differently, it's kind of interesting. So just a couple of weeks ago, I gave a talk on what I regard as the importance of failure in the creative process. And so when I look back at what at the time felt like failure, yeah, sure, we didn't succeed at it, but the lessons we learned were really vitally important. So Simon talked about having a sort of naive view of what the application space was going to be. So if in 2005, when we first started talking about this, if we'd have been just given the money then, I don't know how well we would have done, because our thinking was quite naive. The field wasn't ready for it because we had to make people think about these things with more belief than they were ready for at the time. And then the other part of this, of course, is, can you imagine the computer trying to deal with the terabytes of data this thing generates with the computers of 2005? Joke. So I actually think that those failures to get funding, all those grants that we wrote that weren't successful, they very much, in retrospect, contributed to the final success of the project. I think if we'd have got the money just given to us in 2005, we'd have ended up in the same way that the P39, the SHR 92000 did, really. We'd have probably made one scanner that would have sat in the basement of the engineering building in Tupper Hall at UC Davis. Maybe we'd have scanned 10 patients and that'd have been it. But with the time and the talking and the discussion and the maturation of the concept and the maturation of the field as well, when this thing finally hit the market in 2019, people were ready for it. And we thought that maybe there'll be 1 or 2 of these as international facilities. But now there are 50, 60 of these things dotted around from different vendors across the world. So instead of it just being like a giant telescope that you book time on and people come from all over the world to use, this is now, it's a whole field now. And I don't think that would have happened had we succeeded early. So it's easy perhaps for me to say this now, but if I was to rewind time, I probably wouldn't recommend that we do anything differently, actually.
Everything happens for a reason.
But we'll see.
Yeah. Everything happens for a reason, that's right.
So I guess just moving on as a segue from your answer there, Ramsey, if we move forward to present day, has the global uptake of total body PET in any way surprised you?
Yes.
100%. 100%. I mean, as I say, when we wrote the grant, we imagined building 1 or 2 of these things as initially as a national facility and perhaps as an international facility. Because, I mean, we kind of trapped in our own, with the problems of the sort of Stockholm syndrome I talked about earlier. There's no way they're going to buy more than 1 or 2 of these things, right? I mean, that would be crazy. So we'll have one or two of these things. We'll have a center of excellence. People will come, we'll do projects for them, and it'd be great. And it would have been great, but a bit of a waste of an opportunity, I think, because no one group can possibly do all the things that you can do with a scanner like this. So our first hint that perhaps things might be different is when we were talking to our industrial collaborator, and we were having dinner with the director of the company, and he was talking about $1 billion market. And we were like, what? You must be joking. But if you think about it, we're not at $1 billion yet, but like if there's maybe 50, 60 sites, there's several $100 million been sunk into this technology already. So we're not that far off. We're in the right ballpark actually for $1 million market. So yeah, I would never have dreamed. I'd say that's the kind of vision I think that you know, an entrepreneur needs to have. I'm not really one of those. I never imagined that we could have had $1 billion market here. Well, I mean, Simon, what do you think?
Yeah, so I think I'm quite surprised by the clinical uptake and how rapid that's been, given that, you know, the cost of these systems is obviously very high. And yes, you can have higher throughput, but still, you know, if you look at most of what we're doing clinically, it's doing what we could already do somewhat better, which is great. But in a lot of cases, cutting down the scan time, which is lovely for the patients, obviously, particularly if they're in pain, you know, having shorter scan times is really good, or cutting down the dose a bit. But I would say the clinical use in general has hardly been revolutionary, which is why I'm a little surprised that the uptake has been so quick and so high. On the flip side, on the research side, I had really high hopes that we would be doing fundamentally new science once we could image the whole body. And to date, you know, I think this is taking longer than I'd hoped. I'm not seeing those papers coming out that are really taking full advantage of the total body dynamics to elucidate something new about the biology of health and disease that gets into these really high-end journals. And that's, to me, a little bit of a disappointment so far. Perhaps I'm just, you know, asking for too much too soon, but I think the opportunity is there. This is a unique tool. I don't think we're using it yet to its full potential.
So of all of the different ways that you've sort of had oversight of people using total body PET, are there any applications that have particularly caught your attention?
Well, there's a couple of things here. I don't think we realized how important this high sensitivity was going to be for doing immuno-PET imaging. That wasn't really, I don't think it was even part, I can't remember if it was even part of the original grant proposal, but it's a game changer because when most of the immuno-PET studies are done with zirconium-89, which the dosimetry for the patients is very poor. So it has long half-life, 3 days. You need them if you want to image these antibodies over a long time. But it having a long half-life means the dosimetry profile is bad. On top of that, only 20% of the decays result in positron emission. So most of the dose is going to the subject and not resulting in usable signal. And as a result, you basically have to inject tiny amounts of activity and then you get very bad signal. And when you image that on a regular PET scanner, you get barely understandable images. And if you want to do really meaningful science, you probably have to do population-based analyses, and it's difficult to do really good stuff on a single patient in many cases because the images are just too awful. But when you put a patient like that on this scanner, the images are actually pretty good. They're so good that you can even do dynamic imaging and kinetic modeling on there, which we had never imagined that you'd be able to do. So that was sort of a bit breathtaking. I think the other part of this is I'm starting to see now that the methods are starting to become in place where maybe we've got a fighting chance of curing Simon's disappointment. One of the issues here is that all our mathematical treatments, all our processing techniques for 50 years have been predicated on really just imaging a single organ. Or if we were going to image the whole body dynamically, we were going to have to sweep the body to and fro and therefore have terrible temporal sampling. Yeah, I mean, the amount of sampling you can do, the amount of points on your time activity curve are very sparse if you have to push the patient to and fro in a scanner that's only 15 or 20 centimeters long. So I don't think we really didn't have the tools actually to do proper dynamic analysis. And we're starting now to get the tools that allow us to do that. We're starting to build kinetic models that really depend and are tailored to different organs and can be applied to the same whole person data set, but with the right thing being applied to the right part of the body. And we're starting to develop techniques which can take advantage of the high temporal sampling. If you, like typically in PET, you basically said, okay, well, you can't have frames that are shorter than 10 seconds because the noise is too high. And if you have frames that are short, that are about 10 seconds long, that means you're actually missing a lot of the early part of the change as the PET radio tracer goes through the body. Now, with this scanner, we can go down to 1 second frames. And now that means we actually not only can, but we actually have to start throwing away some of those assumptions that we had to make to deal with 10-second frames. And that suddenly opens up an enormous range of new possibilities and new biomarkers that we can get. We now, for example, can get quantitatively measure perfusion in all organs of the body, all at the same time. That wasn't really possible, and the math wasn't even there for us to do that until quite recently, and it said it wasn't easy to validate. So now we're starting to get the tools, I think, that will allow us to, I hope, let Simon sleep better at night, because hopefully we can start to use this thing for what it's really supposed to be good at, which is total body imaging.
Do you have any comments to add there, Simon?
Well, Ramsey picked the two things I was going to pick as well, so that shows that it's good. So I mean the immunopet for sure. I mean there we are actually already seeing some initial good results from several groups now, so that's exciting. And I will point out that that we're able to do these studies now in healthy volunteers. That was never possible before because of the dosimetry, and the amount that we're actually injecting, or the amount of positron emitting signal there is coming out of the whole human body, is about the same as we have in a mouse study. So the fact this is diluted across the whole human body and we still get great images is pretty amazing, as Ramsey says. So it really has revolutionized that area. And then I completely agree with the dynamics. I mean, yes, we can go down to 1 second frames, which is really great, but I think there's a prospect of going even faster. We've shown that as sort of proof of concept. It's computationally a tour de force to do it, but once you get down to about 1/10 of a second, which you can still get reasonably good images even there with some tricks. Now, as you watch the bolus you inject go through the entire cardiovascular system, there's so many different parameters that you can get about cardiovascular health for any tracer that you inject, as long as you do fast dynamic imaging at the beginning. And perfusion is one of them, but there's many other things you can get as well. So I think this realm of ultra fast imaging as you're injecting can open up a lot of interesting things. And then as we think about integration with systems medicine and network medicine, about how we look at interactions between different organs and how we build the models for that, how we integrate with other disciplines producing very different data sets. So think about the omics, technologies that are, deep phenotyping, measuring lots of different things, but only from a blood sample, or if they're very lucky, a biopsy sample, so no spatial information. Wearables, which produce fantastic data over time, but again, measuring a relatively small number of things and only from one location in the body. And then how do we integrate that with total body PET that gives us 1 million different spatial points in the body? Dynamics of a tracer. But of course, we're only going to get a relatively small number of measurements, and certainly a relatively small number of people. So our cohorts are small. How do we find ways to integrate all of this so that total body PET can really contribute to this emerging field of systems medicine? I think that's one of the big challenges and exciting opportunities for us.
So yeah, you've just mentioned the potential move to ultra fast imaging and integrating with other disciplines, and potential for personalized medicine and biobanking. Obviously, there are numerous game-changing benefits that are already available with total body PET including the reduced scan time and the lower radiation doses, and revolutionary opportunities to see the whole body processes and signaling between the different organs. But I think you've sort of started to answer this, Simon, but I think maybe looking a little bit further into the future, what do you see as the most exciting opportunity for the future of total body PET, maybe in the next 10 years or so?
Well, I think it really is on the application side. It's really, addressing the human body as a whole. We know so much of what goes on is an interaction between different tissues and organs. Even diseases and processes we thought were the domain of a single organ, like Parkinson's disease in the brain, we now know that the gut-brain axis is very involved. The immune system is everywhere. So the ability to watch how the immune system responds to a challenge, whether that's an infection like COVID, for example, or cancer, I think the opportunity is incredible. And we just have to grasp them, and we have to integrate with people who probably never heard of PET right now. We've got to find these people who are the true thought leaders in some of these other areas of systems medicine, and get them enthused and get them engaged. And that whole systems medicine field is a pretty young field, rapidly growing, lots of questions about how it's going to contribute, how to analyze data, how to incorporate different kinds of data, and we need to sit at that table, and we need to find how to get a seat at that table, I think. So for me, the applications are really the key. Technologically, there's still some fun stuff to do as well. These scanners will continue to get better. We're already seeing the second generation systems. The obvious thing to push on is the time of flight performance, which now in the latest systems is down to 200 picoseconds, which is a few years ago everybody would have thought that was incredible. What I'd love to see is, can we get that down another order of magnitude, down to like 20 picoseconds? If we can do that, then every time we detect an event, we know exactly where it came from, well, within 2 or 3 millimeters. We don't need image reconstruction to tell us that anymore. So then we're getting the maximum information content essentially from every event. And I know we'll get there at some point. I don't know how quickly we'll get there, but we will. And so technologically, I see that will continue to happen. But there's only going to be investment if the applications, either clinically or in research, justify it. And so that's why I think we have to lead with exciting new applications.
Ramsey, would you like to add to that?
No, I agree. I think, in a sense, these two things go hand in hand a little bit. So I talked about the new analysis tools we need to analyze all the body. And how you have different parameters for different organs. Well, okay, now you've got more parameters to fit. Well, that's a bit of a problem, because do you actually have, in spite of the fact that you have enormously more data with this thing, do you have enough data to fit all these new parameters that you just discovered? And of course, if you can push the time of flight down, then the data quality goes up, and it does become possible to fit these additional parameters. So I'm still hungry for a better quality total body PET scanner. There's a big movement towards having sort of cheaper total body PET scanners, and I think that's very laudable. But without the applications, I'm not quite sure what the win is, actually. One of the things that's been occurring to me, actually, is that regular PET scanners have been getting better and better, and that's great, and in no small part, actually, because they've been pushed by this thing. But what that means is that the incremental value of having just a long scanner compared to a very, very high performance regular length scanner, if all you're going to do is image faster, do shorter scans for the clinic, or image with lower dose, or do more throughput, if all you're going to do is that, there's going to come a point where actually a 35 or 50 centimeter scanner with 100 picosecond timing can kind of do everything you need. And then simply the justification of, well, I can image my cancer patients faster, is a little thin. Really, if we want to see a great future for total body PET, we have to develop these total body applications. That involves, as Simon says, bringing more people in, but also it's not science fiction or wishing for the moon here a little bit. If I may, if we've got time enough, if I may, just have a little think about what you can do with that humble little molecule, fluorodeoxyglucose, with one of these scanners. So now you can look at glucose metabolism. We've known that for many, many years. You can look at inflammation in the liver. Think of all those people who've got fatty liver disease that we're very interested in knowing whether their livers are inflamed or not. We can do that without a biopsy now, just with using FDG. We can measure perfusion in every organ. I talked about that a little bit before, but that means that, like, for example, if you've got diabetes and you've got poor circulation, you can start to maybe probe that with your humble FDG molecule. You can look at, potentially, we might be able to look at myocardial perfusion as well. We can look at cerebral perfusion. We can look at, and this again results from this sort of new mathematical approach, not just perfusion in the brain, but the effectiveness of the blood-brain barrier. And we're not talking about massive breakdown like you get in cancer, but just an intact blood-brain barrier. How well is it function? Turns out that can be correlated with disease state. And again, just with FDG, $100 a dose, you can measure that. And on top of that, odds on, I think we're probably going to be able to look at kidney function as well. We know that kidney perfusion certainly is related to kidney function, and it's very likely that we can actually start to look at filtration rates and so on and so forth. Again, with this simple, humble molecule, we can look at least qualitatively, we can look at the immune system in the lymph nodes. They're a little bit small for the scanners that we've currently got, but you can certainly see qualitative change. For example, after a myocardial infarction, you can see the lymph nodes lighting up as you get systemic inflammatory response. And you can also now, by doing clever things with sort of dual energy PET CT, using 511 kV PET data and the CT data, you can do tissue decomposition and work out what fraction of the bone marrow region is actually red bone marrow. And so you can quantify bone marrow response. So think of all of those things. That's a huge amount of information about the body available in one patient from one scan. So treating that holistically, that's a tremendous opportunity there. We don't have to wait for that. We can get going on it right now.
That's a really, really interesting insight. Ultimately, treating people holistically could improve outcomes for a huge range of diseases. But it's really interesting that it seems that this kind of comes back to bringing the right people along the journey, engaging the right people, but also combining that with, as Simon said, the tech fun and developing the technology to that point as well. So thank you very much, Simon and Ramsey. With that, that's the end of our structured questions. We're now going to open the floor to our audience, but I'd really like to thank you both for this excellent discussion. And I'd like to hand over to Professor Meikle to lead the Q&A session.
Thank you, Alex, and thank you, Simon and Ramsey. That was really enjoyable and very, very interesting to hear all those insights and to hear a little bit more of the story. I thought I knew most of it, but there's always little bits that you pick up when you hear you talk about it at length like that. So thank you very much.
You're not going to make us sing, Steve, are you?
I might get you to demonstrate how you can compose a song in real time. That'd be great. Take it away. Look, so there's a couple of questions in the chat, and one of them I think you've really already kind of answered, but I'm going to put a bit of a twist on the question. So it comes from somebody you both know very well, Dale Bailey, and he's asking, what is the next big challenge for the field? So I'll let you have a go at answering that if you want to add anything to what you've already said about, for example, the time of flight challenge. And then, I'll let you have a go at that, and then I'll follow up with another question. So, Simon, do you want to go first?
Yeah, I mean, I think I said what I wanted to say already, in the sense that I think, if we look at the research area, which I think will ultimately drive new clinical applications, why it's really important to invest in that area. I'm not seeing as much activity as I would have perhaps hoped. I just got back from the latest Total Body PET conference, which was last week, which was, good. There's lots of good work going on, but I didn't see lots that was really stunningly new on the research side. And I didn't see a lot of new people who had sort of been attracted into this field and are starting to use this as a tool. And so I think it does come back to answer Dale's question, how do we integrate ourselves into this emerging discipline of systems medicine where, we're trying to understand the body's interactions both in health and disease as a whole. We have a bunch of technologies already that are sort of ingrained in that area and producing data, but we are coming along with a new technology that brings very different kinds of data, but very unique data. And how do we get into those communities, engage, and start to design the kinds of studies that will provide preliminary data to sort of demonstrate what total body PET can do, where we're not, we're really taking advantage of the power of seeing all the organs at once. We can measure what's going on in all the organs at once. And I think, one of the possibilities for what we may need to do is to sort of go to what I'll call challenge or activation paradigms, just like we did in the brain in the early days of PET, and which we do with fMRI, functional MRI now. Rather than just making one measurement, we try and perturb the system while we're taking data. So obviously there's lots of things that can change perfusion, change glucose metabolism. It could be a pharmacological intervention. It could be an environmental intervention. It could be a physiological intervention. There's lots of ways to challenge the body and look at the response, because then each subject serves as their own control. And that's one of the things that we measure with PET are highly variable from subject to subject, which means if you're going to do group-based analysis, you end up needing very, very large cohorts, which is incredibly expensive. So I think we need to develop these kinds of paradigms with all the modeling to go with it, because now you're talking about non-steady state conditions. And Steve, I know you have an interest in this particular area. And so I think there's a lot of value to be derived by thinking about those kinds of protocols.
Okay, thank you. That's great. That's very interesting. Yeah, as you say, that's something that we're very keen to explore. And that kind of does segue into my follow-up question, which is, I think one of you mentioned earlier on that, of course, our small animal PET systems that are available now are also total body PET, right? And I don't think there's been as much of a fuss made about that, really. And I don't see a lot of research centres taking kind of full advantage of the total body capabilities of both their small animal PET systems and these total body human PET systems. So I guess the question is, and perhaps that example of challenge studies, pharmacological challenge studies might be an example of this. Where do you see small animal PET fitting now? Has its role changed? Because if you think about it, you can because of the very low dose, we can very easily go into humans quite quickly. So where do you see the role of small animal PET in the field as a translational tool? What sorts of studies should we be exploring there before we then translate them to first in human studies?
You want to take that, Ramsey?
Yeah, so you might be better placed to take it than I, Simon, but maybe I'll make a comment or two. I mean, we do still live in a fairly tightly regulated environment for human studies. So of course, in the development of radiopharmaceuticals, the preclinical model is still going to be strong, simply because the burdens of getting your radio tracer to the point where you can inject it in humans still remain very high, and there's a sort of valley of death between development and getting it into the place where it's regulatory considered safe to inject into humans. It's quite steep. It's quite a deep valley. So of course you can prototype stuff in preclinically there that you couldn't otherwise do. So there's that, and I don't think that's going to go away anytime soon. There are a few challenges, of course, with the total body paradigm, and I think we need to ask the right questions. Because these small animal scanners, they don't have the relative spatial resolution that a human scanner would have, right? So, even if you have like a 0.5 millimeter, imagine a really advanced, very powerful small animal scanner that at 0.5 millimeter spatial resolution, 20% sensitivity. Sensitivity would be up on a par with what you could get on a scanner like this, but the resolution compared to the size of the organism would not be. And so that stops you doing some of the studies that you could do in a human, in a mouse. So then perhaps you need to be looking at the kind of studies you can do. You can look at organ-based kinetics, perhaps. Looking at lesion-based kinetics, that's maybe a bit tougher. Looking at very small organ-based kinetics, tougher because of the partial volume effect. But organ-based stuff you could do. And so looking at things like interactions between different organs and looking at how they, as a result of, say, for example, as a challenge study like Simon is suggesting, that should be possible, and you should be able to do some prototyping there. So, I mean, metabolic stuff, nutritional stuff, actually is quite hard to do in humans. You can do it, but it's practically quite tricky to do. Much, much easier to do that preclinically, at least to prototype the protocols. So maybe that's a place we could start. Simon, perhaps you'd like to add something to that.
No, I think they're all good points. I mean, another advantage in the preclinical setting is that you can often get tissue for validation, of course, which is, is an important thing to consider. But I was going to say exactly what Ramsey said regarding the resolution challenge. It does limit some of the things you can do. And so maybe there's actually a place for pushing the small animal imaging technology a bit, because although there are some limits for sure, I don't think the current commercial systems are as good as they could be. They're obviously quite cost constrained, and so I'd love to build expensive things, as you've probably gathered by now. So we could probably build the world's most expensive small animal PET scanner as well, if you wish, where we really push the resolution as far as we can go, very good sensitivity. And I think that's what you'd need. You'd really want a high-end tool to do some of these things. But as Ramsey said, there's a subset of things you can prototype, and I think good centers will probably find a way to bridge across the human and the small animal, and don't forget large animals. There's a lot of early PET research was done in larger animal models. Then it's quite challenging sometimes, but the resolution issues largely go away then. So if you have access to non-human primates, to dogs, to pigs, depending on what you're trying to model, that could be another approach as well.
Yeah, very good.
Maybe go back to Dale's question about the next challenge to the field. I think there's something I'd like to add actually, which is I think the next challenge is partly sort of regulatory and administrative, and it's partly intellectual, and it's about, I feel the next challenge is to get away from blobology in cancer. So PET has done very well in terms of integrating into the clinical management of cancer by virtue of the fact that we can see hotspots where cancer is and do some quantification on them. But the application space for a scanner like this is far, far greater than that. And so we need, I feel like take the fatty liver disease issue, right? So that is a massive, massive problem across the developed world and across a large fraction of the developing world as well. Now, the idea of using PET for that is kind of unthinkable right now. Nobody would dream of it, except possibly to do a few research studies and maybe people get a PhD out of it and sell it and publish a few papers. But to actually get it out into the clinic, no, that's just not on the radar. And I think we need to get these things onto the radar, because that's where the real benefits for the really new benefits for alleviating human suffering are. They're in some of these chronic diseases. So in order to do that, we need to make this scanning, well, first of all, we need to develop and prototype the protocols where this can change management. Having done that, we then need to broaden its applicability. That probably means making the scanning protocols easier. It may mean making scanners cheaper. In order to, because otherwise, how do you meet the need? In the US, there's 60 million people with fatty liver disease, right? If you're going to, and probably 10 million of those have an inflamed liver. So if you're going to scan all those people, you need to make PET scanning an awful lot easier than it currently is. So there's a challenge, I think, for us if we're really going to take advantage of the new methods that we're developing here. But it's maybe recasting how we think about these things being used in the clinic as a challenge.
Yeah, and I think again you've got to find some believers, right, in these other clinical areas. We know from our own experience in trying to get funding in some of the non-oncology areas that Our review panels, have a pretty dim view of PET in general as being, the radiation dose always comes up as a big issue, and limited spatial resolution often comes up, and so we need some visionaries in some of these other areas. And Ramsey just mentioned one big area, but there's quite a few others as well. There's the autoimmune diseases as well, another big.
Seems like we've been hit by Davis internet there.
Oh, yeah, cost.
Yeah, we just.
Yeah, am I back now?
Yeah.
Yeah. So cost and radiation exposure tend to be things that review panels are struggling with. So how we communicate that we can do things effectively and safely in people with chronic disease rather than elderly patients with life-threatening cancer, that's a major challenge for us, is how we break in to those other communities.
Yeah, and look, we're coming very close up to the hour, so we'll have to wrap up soon. There are several more questions that I'd love to ask you. I think we've got time for just one more, which kind of follows on nicely from what you've just been discussing. It comes from Tao Sun, and he makes the point that when you think about all these total body or long axial field of view PET systems around the world, they're almost all installed in clinics, very clinical centres. And so he asks about that challenge of competing time for research when these systems are in clinical centres. And I guess the question also is, if you think about reaching out to those thought leaders in other disciplines that are not currently doing PET, can you do that in a clinical centre, or do we need some sort of landmark centres that are just focusing on the research alone? How do we do this?
Yeah, it's a good question, because I don't think the research volume and funding is there yet probably to support scanners full time for research only. I think that's a hard sell right now. There are quite a few scanners that have a joint research clinical mission like ours, and I believe that your scanner there also does as well. So that's good, because there is protected research time, which is important. So it's a bit of a chicken and egg situation in a sense. We would, I think, Ramsey, you can correct me if I'm wrong, we would do more research on our scanner if the funding was there to do it. But we've struggled in some of, as I said, the non-oncology areas. It's not been easy to get funding for some of these ideas, not at the level you need to run lots of patients through.
Thanks, Simon. And by the way, we might get cut off because we're coming right up to the hour. So if you want to add anything, Ramsey, please do. But I just want to make sure I don't miss the opportunity to thank you both very much for your generous time for this webinar. Ramsey, did you want to add anything?
It's just a great pleasure to have been here, and really appreciate the invitation. And it's great to see you again, Steve, and nice to see you, Alex, and to talk to you all.
It was wonderful to have you. Thank you.
I think we'll wrap it up there then. And Alex, I'll throw back to you to wrap up the webinar. But yeah, thanks again very much. Really appreciate your generosity with your time, Simon and Ramsey. It's been really insightful and very enjoyable.
Fun.
Over to you, Alex.
Thank you very much for joining us today. Our recording of this webinar will be available on the National Imaging Facility website. As soon as it's available, we will advertise it. Thank you, everybody.
Bye-bye.
Thanks, and see you guys soon. Bye-bye.